Yes, welcome everyone today's Office of the Chief Scientist Science Seminar series. I'm Andrew Paul, a Senior Science Advisor with the OCS, and we'll be hosting this afternoon's seminar. On behalf of the whole OCS team, welcome to today's event. I'd just like to start off in the spirit of reconciliation, acknowledging that Alberta is on the traditional territory and treaty land of the Cree, Blackfoot, Nakota Sioux, Iroquois, Dene, and Saulteaux nations, as well as the homeland of the Métis. Wherever you might find yourself today, I encourage you to recognize and acknowledge the relationship that First Nations, Inuit, and Métis have with the land which we all live, work, and learn on. These science seminars provide an opportunity for us to gather and exchange knowledge on interesting science from across Alberta in an open learning and sharing environment. I encourage you to enter any questions you might have or comments at any time into the chat box in Teams anytime during the presentation. To maintain our call quality, your video and audio is disabled, but after the presentation, I will read out the questions in the order that they were received. Sometimes those questions get a little, or we don't have enough time to go through all of them. Any that we aren't able to address today will certainly forward to today's speaker to respond and look at. And with that, I'd like to welcome our speaker today is Dr. Patrick Hanington, who will be presenting Spinning Tails, Exploring Whirling Disease in Alberta. Patrick is a professor in the School of Public Health at the University of Alberta. He and his research group work on developing and implementing DNA-based tests for priority organisms found in aquatic environments. Their research often combines large-scale spatial and temporal studies that rely on community-based partnerships and combine these with cutting-edge molecular biology. Recently, Patrick has been focusing on the detection of aquatic invasive species and species at risk using environmental DNA. And with that, Patrick, I'll turn things over to you.
Great, thanks, Andrew, and thanks for the invitation to present today. It's a real pleasure to be here. I'm excited to talk about our work on whirling disease, and I wanted to start by giving everyone just a bit of a background on whirling disease before I jump into this stuff that we've been doing. And as I was doing that, I put together a little sort of timeline of whirling disease in Alberta, and we're actually coming up on the 10-year anniversary of whirling disease detection in Johnson Lake and Banff National Park, which was August 2016. So about a year and a half away from the 10-year anniversary. And I think sort of theme in my presentation is going to be that this has moved very quickly, and I think we've done a pretty incredible job actually learning and trying to understand more about whirling disease in Alberta in a specifically Alberta context. So as you can see here, I just have a series of snapshots from news headlines. This was one of the headlines when whirling disease was first discovered in Johnson Lake, and it was followed up a couple of years later with what I think is one of the more incredible efforts to control the parasite in Johnson Lake, which was when they actually drained the lake to try to eliminate the parasite. And we were sort of peripherally part of some of that work, not the draining itself, but trying to understand more about whether the parasite had actually been removed from the lake after the draining process. And then more recently, we've seen whirling disease jump over the Rocky Mountains and has now been detected in British Columbia. It was detected there a couple of years ago in the national parks and has now moved outside of the national parks. And most notably last summer, there were restrictions for boat access and in some cases access to some of the national parks where whirling disease had been detected as a means to try to help with control efforts and also to minimize the chance of spreading the parasite. So this fish infection has really caught the headlines and has also really been a priority in Alberta, and now I think it's becoming a priority in British Columbia because of the effect it can have on some of the fish that we really care about. Just so that everybody's oriented to what it actually does and what it is, whirling disease is caused by a parasite called Myxobolus cerebralis, which in my talk I'll either call it M. cerebralis or sometimes MC to save some space. and my tongue. It's an invasive parasite in North America, which kind of fits with the theme of us working on invasive species these days. It's actually probably the first invasive species that we really started working on in earnest with respect to our program on environmental DNA. And it originates in Europe. It is the causative agent of whirling disease in salmonid fish, trout, whitefish in Alberta in particular. But it's important to note that not all salmonid fish display symptoms equally. And so some display very serious disease like rainbow trout. Most strains of rainbow trout display the very classic symptoms of whirling disease, which includes this blackened tail, which originates from the infection damaging the cartilaginous tissue in the tail during the developmental phases of the fish while it's a juvenile. A shortened operculum, which you can see here, often neural damage as well, comes along with this in the brain, and the forehead is often sloped in a little bit of an unusual way. And so fish that display severe whirling disease often display these types of phenotypic characteristics. And then other fish, other salmonids, can be infected by this parasite, but maybe don't develop disease. And that has implications for transmission, but won't be a real focus of what we're going to talk about until later on in my talk. It's important to note, because we get asked this question a lot, no disease is caused in humans. And so if you eat a fish that is infected with whirling disease accidentally, or you are exposed to the parasite in the water, you don't have anything to worry about. And just so you can see, this is an image taken from one of the studies actually that was focused on fish in Alberta, James et al. From 2021. And in this image, you can see a cross-section of the spine of a normal cartilage possessing fish. And then you can see here one that has a severe parasitic infection of Myxobolus cerebralis. And you can see that sort of normal healthy cartilage tissue is replaced by these myxospores, which are one of the stages of the parasite. And this is really what leads to this blackening of the tail during the developmental stages of the parasite. What's also really interesting about this parasite is it's actually most closely related to the cnidarian, or the jellyfish. And this is most obvious if you compare the morphology of the stinging cells in a jellyfish, these cnidocytes and the nematocysts, with the polar capsule of the myxospore, where you have this coiled structure that essentially shoots out to help the parasite or the cnidocyte either sting an organism that's encountering it, or facilitate penetration into the host for the parasite. And so I tell you this because I think it's cool, but it's also made it fairly simplistic for us to develop DNA-based tests for this organism, because there's only a handful of myxozoans that we know of in Alberta, and there's only a few more organisms that relate to jellyfish closely enough that the DNA of Myxobolus cerebralis can't be used to distinguish them. So it makes it quite simple for us to design an eDNA test for this organism, which I think is really useful. In terms of the life cycle of this parasite, I think I've focused on the fish so far, and so we'll start there. If you have an infected salmonid fish, and these are the myxospores that are in those fish, the myxospores can be passed from an infected fish, and in particular are released when an infected fish dies. And those myxospores are usually heavy, and they'll fall into the substrate and the sediment of the water body that fish was in, the stream, the lake. These myxospores are consumed by an oligochaete of the genus Tubifex, usually Tubifex. These little pink worms live in the substrate, and they eat the myxospores and then become infected. And after usually a few months following the consumption of those myxospores, they'll release another spore stage called the actinospore, which is abbreviated as a TAM. It stands for triactinomyxon. And the TAM, when it's released by the worm, it comes out in the worm's poop, and it inflates like a balloon when it hits the fresh water, and becomes pelagic, and it floats around in the water column until it encounters a fish. And then when it does encounter a fish, it'll penetrate the skin or the gill of the fish and initiate the next stage of the life cycle in the fish. And so there's quite a few stages of this parasite that are important for us to keep track of, and I'll touch on each of them, the environmental, the fish, and the worm, throughout my talk today. Before I do that, though, I just want to emphasize why we care about whirling disease. I've shown you the phenotype that can happen in a susceptible fish like a rainbow trout, but I want to emphasize the significance that this parasite has at a population level. And so, in the United States, whirling disease was first detected in Pennsylvania in 1956 at a fish hatchery. Since then, it has moved across the United States and has been detected in 25 states. And in some of those states, Colorado in particular, fish populations have been incredibly hard hit to the point of in some water bodies and watersheds up to 90% or higher reduction in the fish populations. And this is important for several reasons, but I want to specifically focus on the story in the Colorado River here just to emphasize the impact this parasite can have. So the parasite was first detected in Colorado in 1987. And on the graph to the right here in this Eric Featherman's paper from 2014, you can see it's a three-dimensional graph where we have the number of fish that were observed along the y-axis. We have the length of those fish that were collected in length categories across the x-axis, and along the z-axis, we have date with the earliest date starting around 1984 and then moving to 2008. And as you can see, if we look at the back of this graph, there's a reasonable size distribution and good recruitment from the small fish into the large fish year to year, up until sometime in the sort of mid to late 80s and maybe early 90s. And around the early 90s, what we see as you move forward along this axis, the z-axis, is basically a complete loss of the small fish in subsequent years. And so you can see there's almost no young fish in any of the years past sort of the mid 90s. And that then the consequence being that they don't recruit into the larger size classes, and you have a complete collapse of fish in this area. This is in the upper Colorado River, one of the hardest hit areas by whirling disease. And so this graph is one of the more sort of noteworthy ones that we use when we talk about whirling disease and the impact it can have. And as you can imagine, this graph, having just come out in 2014, was circulating, making the rounds when we first detected whirling disease in Johnson Lake and Banff in 2016. And up until 2023, the parasite was confined to Alberta, so we were the only province in which the parasite had been detected, up until 2023 when we detected it in BC. And one of the biggest concerns when it was detected, obviously, was that something like what happened in Colorado was going to happen in Alberta. And it was pretty much right away that my group and I got asked to participate in some of the early whirling disease projects, both by Alberta Innovates and then Alberta Environment, and at the time, Alberta Environment and Parks, now protected areas. And one of the tasks that we endeavored to try to help with was to simplify sampling and testing methodologies for whirling disease. As you can imagine, with a parasite like this, where there's these two environmental stages that look quite different, and there's a stage within a fish that we have to actually sort of do lethal sampling to detect in most of the fish, and there's these worms, we thought that a DNA-based testing approach would be an ideal way to simplify testing across all those different types of places where you might test. Whirling disease testing from an environmental context is quite labor intensive. Water sampling for the triactinomyxon stage, those TAMs, is very laborious. Thousands of liters of water have to be filtered to collect TAMs. You need a trained eye to be able to identify the parasite within tissues. There's often a sort of a complex histological process that has to happen where you have to digest the tissues and then look for the spores. And we know from looking at fish that can be infected with different species of myxozoan that the myxospore morphology, which is shown on the right here, can be very similar across different species. So you need somebody who knows what they're doing to be able to key out that this one here is Myxobolus cerebralis and these other ones are different species that are irrelevant to your worry about whirling disease. And then finally, obviously, this parasite could be impacting fish that we care a lot about, at-risk species of fish, for example, like Athabasca rainbow trout or West Slope cutthroat trout. And you don't want to have to go into those populations, which are sensitive as they are, and electrofish a bunch of them to find out whether or not they're getting infected with whirling disease. And so we thought that a DNA-based approach that allowed us to sort of go and unify around a single methodology in all of these different areas and not have to kill fish to be able to test for the parasite was an ideal way to do it. So I want to talk to you about that story first, and I want to tell you about some of the progress we've made since then using that test, and also trying to understand more about the parasite in Alberta. So one of the things that I want to focus on first is just that we know that the environment, the tube effects, worms, and salmonids are important dimensions to transmission of whirling disease. And I'm going to tell you a story about each of these different areas as we progress through my talk today. It is all going to be predicated on the work that we did right at the very beginning, which was work that was undertaken by Danielle Berry, who is actually now nearly finishing her vet degree at University of Calgary. But Danielle was sort of the champion during her master's thesis project in the lab to develop a DNA-based environmental monitoring test for M. cerebralis. We published that test and some of the work we did a while back, focusing on worms, environmental detection of the TAMs, and surveying histology and DNA extracted from infected fish. And this work sort of formed the foundation of all the work that I'm going to present to you for the rest of my talk. And it was really an incredible joint effort between our group and Alberta Environment and Protected Areas, which coordinated a 3-year sampling program throughout the east slopes of the Rocky Mountains, collected over 5,000 samples from almost 1,000 different locations shown on the map here over 3 different years, highlighted by the different colors of the dots. And from each of those locations, samples were taken that included tube effects worms or oligochaetes generally. We weren't good at identifying tube effects at that point, so we just collected every worm we could find. Sediment was collected, in some cases water was collected, and in some cases fish were collected. And so we had an incredible data set to start off with, and these 3 years really gave us a lot of really important information. Also on this map, you can see where different watersheds that had been declared positive for whirling disease are found, and I'll show you that in a better sort of case on the map coming up here. But really, what this program led to was sort of a progressive identification of locations in Alberta where we found this parasite, far beyond the just simple Johnson Lake detection. It was far more pervasive and likely was found throughout at least sort of the North Saskatchewan watershed and south in Alberta along the mountains. The map that you would find if you went to the government website looks something like this, where we have regions that have been confirmed positive for whirling disease highlighted in red, and those sample sites that I just showed you are highlighted on these dots. Here you can see where worms and sediment and fish were taken. And then the Canadian Food Inspection Agency, the CFIA, was painting with broader strokes, and they decided that once we detected it and confirmed a fish that was positive in the surveillance that was being done by AEPA, that the whole watershed would just be declared positive. This is an important distinction, just because I'm going to tell you a different story about worms in a minute, and the CFIA had a big impact. We had to first work on whirling disease in Alberta, in particular because I'm at the University of Alberta, and the North Saskatchewan watershed was the last watershed to be declared positive, which meant that water samples and materials that were being collected as part of this surveillance effort couldn't be sent to us at the University of Alberta in the North Saskatchewan watershed without being first disinfected and confirmed that they couldn't potentially release whirling disease in the North Saskatchewan watershed. So there's a lot of decontamination that had to happen in those early days, which had some impacts on what we learned. What we think was noteworthy from this initial surveillance was that the Crow's Nest River, Red Deer River, and the Bow River watersheds were all found to be positive. And ultimately, what we ended up doing was initiating some projects to scrutinize a lot further the Bow River and the Crow's Nest River to understand more about what was going on in those places. The first story I want to tell you here about a specific component of whirling disease focuses on the tube effects worms. And I want to tell you a little bit about what we've learned about tube effects and its role in transmission of the parasite in Alberta, which I think has become a really important part of our understanding of how this parasite is moving through the province. I'll introduce you to tube effects by showing you this video. These are, you can see the little squigglies on the screen. These are all tube effects, and they tend to live in these little colonies where under non-extreme water movement conditions, you can see them poking out and they'll wiggle around in the substrate. These are the worms that will eat up those myxospores. And they can be found sometimes in huge, dense colonies like this. Sometimes they're found much more sparsely, and sometimes you just never find them, but they're very pervasive in Alberta. If you were to take this sediment and substrate and take a scoop of it, and then isolate the worms and try to prioritize the tube effects, you'd be looking for worms that look like this, little pink strands of hair, basically. And the pink coloration is really what gives you at least a way to get down to a subset of worms that you would find in the substrate that would get you close to being always just getting tube effects. However, like I told you earlier, the CFIA made it so that we couldn't get live material in the beginning of the program. So for those first 3 years, essentially, all the worms that we were sent had to be preserved in ethanol to deactivate the parasite. And so when we got worms, even if they were tube effects, they all looked like this, little gray squiggles, which meant that we couldn't use the classic pink coloration to determine whether or not we were looking at a tube effects or not. And so the first thing we had to do when we were focusing on the worms was to actually go in and use DNA barcoding to identify whether or not we had tube effects in different watersheds in the province. That ended up being what I would classify as a deep, deep hole that we went down and barely could get ourselves out of at the end of. At the end of the day, over 5,000 worms were collected as part of the surveillance project, and we DNA barcoded over half of them. And the summary of that is basically that we found 31 different taxonomic groups of worms, essentially 31 different species of benthic oligochaete in Alberta. And as you can see, tube effects was certainly one of them, but there's a lot of other ones in here that we had no idea, and I still have no idea what they really are. For a worm expert out there, though, we've got a lot of information about them. What was very fascinating, though, is if we broke down the worm collections based on the watersheds in Alberta, you can see here that the pink, which represents the tube effects worms, made up basically half of all of the benthic oligochaetes we found in any watershed in the province, which at that time basically led us to hypothesize that any watershed had at least had the worm component of the life cycle for M. cerebralis, which wasn't a great sort of confidence builder in terms of thinking about maybe there's some barrier to transmission somewhere in the province. And so so this was a bit discouraging, but it led us to answering a lot of interesting questions. And one of the places we really wanted to focus on was in southern Alberta in the Old Man watershed, where we found the highest proportion of the oligochaetes were made up of tube effects. Some of the central Alberta watersheds, like the North Saskatchewan and the Bow and the Red Deer watershed, they had a really incredible diversity of benthic oligochaete, which is pretty cool. But in southern Alberta, in the Old Man, it was like just all essentially tube effects and limnodrillis, which are also pink worms. And so it was really difficult for us to identify worms beyond just that they were all pink down there and they could all potentially transmit whirling disease if we just looked at them. So we did a lot of work down in the Old Man to look at tube effects, and I'll tell you that story in a little bit. But one of the things that also came from this big oligochaete study was that when we were looking at tube effects worms genetically, looking at these barcoding genes, what we found was that there were some that were coming back when we would search them against these large online databases. They would come back as T tube effects, but they were very clearly different gene sequences than other T tube effects. And what we ended up doing was finding that if we took those sequences and we used a whole bunch of different genes, including the whole mitochondrial genome of these worms, if we took those different sequences or the mitochondrial genomes and we did a phylogenetic analysis of these worms, we found that there were very clearly 5 different and distinct taxonomic groups of tube effects in Alberta and in these online databases, which reflect the sort of global tube effects population. And one of the things that we found when we started looking at the worms in Alberta and using that qPCR assay that we designed for the parasite we found that most of the worms that were positive for the parasite were these final sort of T3 subspecies. So when we found positive worms, M. cerebralis positive worms, they tended to be T3 worms, this third subspecies. That over that sort of 3-year project with APA, we came up with a hypothesis that it was possible that T3 worms were the main driver of whirling disease transmission in Alberta, which we wanted to try to test. And so we set out to try to answer that question over a few years, and we're actually just wrapping this study up. So it's like a perfect timing for me to be giving you this presentation. So we asked this question: are the T3 tube effects really the primary drivers of M. cerebralis in Alberta? And so one of the advantages of these worms is that they're hermaphrodites, and so we can take these little pink hairs that we can collect. And in a partnership that we did with a researcher at Oregon State University, we essentially created self lines that reflected the T1 to T5 subspecies. So we made lines of each of the different subspecies. And then after we got those lines established, we challenged them with M. cerebralis, and we looked, after the infection had progressed, we looked to see if they were releasing TAMs. And I mentioned this earlier, but the way that these worms release TAMs is they essentially poop them out in these little fecal packets. And these fecal packets are pretty tightly packed little packages of TAMs, but when they reach the fresh water, and you can kind of see it here maybe, the TAMs will start inflating like little balloons, and they'll start drifting off from the fecal packet. And so you can have thousands of TAMs released in these fecal packets over a period of time. And ultimately, what we're looking for is something that looks like this. This is a triactinomyxon of M. cerebralis, with this little shiny bit here being the part that will inject the material of the parasite into the fish. So we set up this study. We challenged all these different lines with parasites. We looked for TAM release after an 11-week period of parasite development, and then we did that over 5 consecutive weeks. And what we found was really fascinating. So this graph here shows us the number of TAMs that were released each week by each of the worms that were in the study. And keep in mind, each of the worms are individual. So each of the worms are in an individual context. And so each of the dots on this graph represent the number of TAMs that were released by a single worm over a week period. And you can see here that these T3 worms, which we had hypothesized may be really important for transmission, they essentially release 10 times or more than that TAMs per worm than the next best transmitter, which would be the T1s or the T4s. And then T2 and T5 don't appear to be able to get infected by M. cerebralis at all. So these tube effect subspecies appear to us as being largely irrelevant to transmission in Alberta. T4 and T1 are possible and can potentially contribute to transmission, but the T3s are certainly the largest driver in a very big way. And so we think this is a really supportive evidence of some of the sort of more anecdotal findings we had when we looked at worms from the field. And I think now we can go back and really try to apply a lens of using the T3 as an indicator of potential for whirling disease transmission in a location. If we took those worms that we collected from this study that Danielle did, and we take these tube effects in the pink and we reanalyze them looking at those different 5 subspecies, what I wanted to show you here is just that in the blue in each of these pie charts is the proportion of them that are T3 worms. And so you can see that with the exception of the Athabasca and the bow, the red deer, the old man, and the North Saskatchewan, more than half of the tube effects that we find are T3s. And maybe even more importantly, in the Old Man River watershed, the worms that we find are only T3 or T1, which are both potential transmitters of M. cerebralis. So to summarize the worm part of this story, I think what I've done here is I've changed tube effects to T3 tube effects. And I think what's maybe important for us to think about in Alberta and maybe in BC as well is looking for these T3 subspecies rather than just looking for any pink worm. And I think that's probably going to be an important thing for us to do moving forward as we get more and more interested in trying to understand where this parasite might move to in Alberta and in BC. Especially in Alberta, why hasn't it moved north past the Athabasca watershed or into the Athabasca watershed? It might be because of the distribution of these worms. So this is something that we're prioritizing now for sort of our next phase of whirling disease research. That being said, I want to shift gears because I want to talk a little bit about the environment as well. The environment is another important component to transmission. The story I want to tell you in the environment component of this talk relates to a publication that came also out of that partnership with APA. This is this paper here, Whirling Disease in the Crow's Nest River. Because we were seeing such high infectivity profiles of the worms, high PCR values in the water and the substrate in the Crow's Nest River, and it's a really important recreational fishing area, it was an obvious place to prioritize work. And this collaboration, which included the people in Alberta Environment Protected Areas, as well as my research team, and then these researchers, Julie and Barry, Julie from Oregon State University, Barry from the Colorado Fish and Wildlife, Parks and Wildlife. This collaboration was really really good. It essentially looked at the worms, it looked at TAM filtrations in the Crow's Nest River, and it set up sentinel cages to assess infection in the fish themselves. And those sentinel cages were set up essentially along the Crow's Nest River. You can see the little dots here at CRR6 all the way to CRR1, and the Old Man River was used as a negative control, where we hadn't seen any whirling disease yet. It's a fascinating paper, and it's worth like reading the whole thing because there's a lot of information in there, and I don't have time to go through everything in the paper, but I do want to highlight a few things from it. First of all, if you look, and these are the sites, the Old Man River and then CR6 to CR1. The first thing I want to emphasize to you is that the percentage of the tube effects or of the oligochaetes that were collected from each of these sites that were tube effects, in some cases, especially CR4, 3, and 2, they were 100% tube effects. In some cases, like CR6, only 28% of the worms were tube effects, but in many of the locations, all of the worms we collected were tube effects. And of those tube effects, I'll jump down. The number that were releasing TAMs, the percentage that were releasing TAMs, was as high as 3% at CR3, CR2 it was 2, and CR1 was 0.5. And when we used that PCR assay, we saw positivity in up to 27% of the worms, which suggests that maybe there were worms that were developing infection but hadn't gotten to the point of releasing TAMs yet. And we saw TAMs released from worms at CR4, 3, 2, and 1. That's really important because then if we looked at each of those locations in a bit more detail, what we found is that CR4, 3, 2, and 1 all had large populations of T3 worms, especially CR2, which was basically 75% of the worms were T3 worms. And those locations, when we looked at the fish in the sentinel cages, CR4, 3, 2, and 1 had, except for CR4, 100% of the fish in those sentinel cages were infected with M. cerebralis by the end of the study. And so there's a really strong overlap between where we were finding T3 worms, where we were finding infected worms, and where all the fish in the sentinel cages were being infected by M. cerebralis. So that was, I think, a really nice reinforcement of our T3 findings. And it's nice because the DNA approach allows us to go back and kind of retroactively look for these things in the DNA samples that we collected. So this has been a really nice way to confirm that the T3 worms are very likely important drivers of M. cerebralis movement and transmission. The other thing that was presented in the James paper, which I think is another really important finding, is the confirmation that TAM released from the tube effects worms is temperature dependent and requires water temperatures between 10 and 17 degrees Celsius. So that's a really important finding on its own. It confirms what has been found in the literature. But what's also really important is that that temperature range overlaps with when the juvenile fish are in the water and thus most susceptible to the parasite. So if you don't have any juvenile fish there when the water temperature is in that range, then you would have very little risk of infection establishing in the fish population. But if you have a lot of juvenile fish and the water temperature is in that range, it becomes a very big problem because then the fish are at their most susceptible when the parasites are being released from the worms. And what that James paper shows is that in most of the sites in the Crow's Nest River, there's between essentially 76 and 96% overlap with this 10 to 17 degree temperature window, and when the fish are the most susceptible to the parasite. This young of the year are susceptible. So the Crow's Nest River kind of became this, essentially, in a bad way, the poster child for whirling disease, in the sense that it had all the criteria: large T3 worm populations, a substantial overlap between it with the window of temperature that M. cerebralis is being released from those tube effects, and the presence of susceptible fish, and susceptible fish being present. And so this study, I think, really emphasized to us that there was a likelihood that M. cerebralis was going to be at very high rate levels in the Crow's Nest River. And I think, as of the time of the publication anyways, Crow's Nest River was the second highest TAM density out of any reported location, the only place being higher being the Gunnison River in Colorado, where they had an incredibly high loss of Colorado River rainbow trout. So then, to wrap up the environment part of this story, one of the things that I think comes out of the James paper is this 10 to 17 degrees Celsius window required for TAM release from the tube effects. And so we've kind of pieced together now that the T3 tube effects existing within water that reaches this range of temperature when you have susceptible fish present is one of these are sort of two really important risk factors that could be used as a way to predict whether or not a water body was going to have potential for M. cerebralis transmission. Obviously, then the final piece to this puzzle is the fish themselves. Our work on this has taken sort of a circuitous route, but it ended up with us partnering with Eric Featherman, who, if you remember, published the paper that showed that 3D graph that I showed you at the very beginning of the talk. And Eric Featherman has been part of an initiative in Colorado to try to create whirling disease resistant rainbow trout. And our interest was trying to understand whether or not we could find out why fish don't develop disease from M. cerebralis challenge. And so we knew phenotypically that we could find in the literature examples of fish that don't develop the blackened tail and the whirling behavior when they get challenged by the parasite. They might still get the parasite and be able to transmit it, but they don't develop disease. And so we were interested in understanding maybe why that was the case. And this partnership with Eric allowed us to work with him and his group on this initiative where they had been developing a resistant rainbow trout strain. And this became big news in the early sort of parts of the pandemic, essentially. So early 2020. Colorado researchers had essentially what they did is they took a German strain of rainbow trout, the Hofer strain, and they crossed it with the Colorado River rainbow trout, which is not resistant, and then they let whirling disease go at it and selected for this strain of rainbow trout that they called the Gunnison River rainbow trout. And the Gunnison River rainbow trout was the focus of our study on trying to understand why some fish are compatible or susceptible to disease and others are not. It's a complicated study because we're looking at the genomes and the transcriptomes of these fish. I'm just going to kind of introduce you to the study that we did, and I'm going to show you one slide that's very complicated, and then I'm going to summarize everything in one little statement. So the study that we asked Eric to set up for us was to essentially take juvenile Gunnison River rainbow trout and put them into either a control group or an experimental group, and the experimental group was going to be challenged with M. cerebralis TAMs, and the control group was not. And then what we were going to do is we set up different time points. We had a time 0 time point, 1 hour post-challenge, 1 day post-challenge, 2 days post-challenge, 4 days post-challenge, and 24 days post-challenge. And from each of those time points, a set number of fish were taken from the control and the experimental group. And we took fin clips from fish, and we isolated their kidneys for transcriptional analysis. The fin clips were for the genome, to sequence the genomes of these fish, and the kidneys were taken for transcriptional analysis. The kidney is an important immunological organ in fish, teleost fish, and so we wanted to know whether or not there was immune genes being transcribed by the kidney in the cells in the kidney at different points in infection. We also kept a group for 5 months that allowed us to confirm that the fish were actually resistant to whirling disease, so they didn't develop any disease symptoms. And then, like I said, this is the result. It's an incredibly complicated study that this is another paper that we're just actually ready to submit now. It's gone through a lot of revision and a lot of work to try to understand what's going on here, because there's a lot of data, obviously. First of all, genetically, the Gunnison River rainbow trout are quite bottlenecked because of the way that they were created, but they do have some really interesting genomic components that we think are maybe aligned with whirling disease resistance. And then what I'm showing you on the slide here are transcriptomic profiles of from the kidneys. And the red is all I really want you to focus on here. The red are just transcripts that are in very, very high abundance in the experimental fish compared to the control fish. And what we found is that at 1 day post-challenge, at 2 days post-challenge, at 4 days post-challenge, and 24 days post-challenge, there were very distinct immunological gene signatures that we could characterize, with the transition being early on, around 1 day, 2 days, and 4 days. There's this very strong innate immune response that's geared towards the killing of protozoan parasites. And then by 24 days post-challenge, we've transitioned more towards what we would consider an antibody-based immunological memory response, so that the fish kind of retain their ability, long-term ability to defend themselves against infection. I'm not going to go into all the different genes and all that kind of stuff. We can talk about that later. But what I want to emphasize is from this study, what we found is that the Gunnison River rainbow trout possesses a genomic region on chromosome 9, which had been previously reported as the whirling disease resistance region. They possess this chromosome 9 region, which is definitely being transmitted or passed on to them from the Hofer fish. And there are also, as part of our analysis, other genomic regions that appear to be under heavy selective pressure in these fish, which I think are worth investigating as potential markers of resistance in the future. And there are also some very interesting transcriptional profiles that we found that highlight the immune response to the M. cerebralis challenge. And they include, like I told you, these very distinct immunologically related genes that I think are certainly important for the immunological component of fighting the parasite. And there's also some very interesting profiles related to neuronal development, which could be around the idea that the fish is still developing the parasite at a low infection intensity, and probably has to accommodate for damage in the tissues, the neural tissues, as a result of that. So these two things made a lot of sense to us, and are things that are the highlights in this paper, which I think we're hoping to submit any day now. We're just waiting for some final feedback. So I want to start kind of wrapping up here, and just kind of emphasize now that in the three circles of my Venn diagram now, instead of just salmonids, I think we have to really emphasize that we need whirling disease susceptible salmonids in this space. And I'm going to talk about that in a little bit, but I think that with the work that we've done in the Gunnison River rainbow trout, we have added to our understanding of the mechanisms of fish resistance to whirling disease development. And now I think we have a toolbox that we can use to start trying to be more predictive about resistance in fish populations. Certainly, I think that needs to essentially be confirmed with a true fish challenge study that is relevant to the fish populations of Alberta. And I think that's something that I'll highlight in my last slide. But to summarize now, I think hopefully what I've been able to, from the whirling disease research we've done in Alberta, we've, I think, shifted the goalpost a little bit. And while my figure hasn't changed too much, I realize, I think we have a much more precise way now of looking at whirling disease in Alberta, knowing that we have a very specific subspecies of 2 effects that seems to be driving the parasite transmission. We know we have a temperature window that we need to be conscious of when we're looking for potential regions where there might be transmission, and when we might want to prioritize efforts to try to ensure that fish are protected. And we know that there are mechanisms that salmonid fish can implement that are at least encoded in the genome and certainly transcriptionally that can help prevent disease development. And there may be some ways in which we can try to use those tools that we've learned about to try to predict whether or not there are specific populations of fish in Alberta that we really need to prioritize conservation efforts for over others that may be just resistant to the disease itself. So in summary, whirling disease has been sort of a rapid invader here in Alberta, and I think that this is something that's really important for us to just understand. It's it's been less than a decade, but almost a decade since we started it, and we still have lots to learn, but I think we've learned a lot. Specific tube effect subspecies appear to be driving M. cerebralis spread and may serve as an indicator of invasion risk, especially that T3 subspecies. And water temperature, I think, also could serve as an environmental barrier to establishment. And at the end here, I think we all know fish can be resistant to disease. I think the term resistant has been a bit of a misnomer here, in the sense that a lot of the fish that we think are resistant can actually still get infected by the parasite, but they just don't develop disease. And that's an important distinction to make, because a fish that still has the parasite can then still transmit the parasite. So we have to be careful about assuming resistance means that we're not transmitting. However, I think it's also important to note that while we have now more information about what makes a fish resistant, we still don't really know the resistance profile for almost any of the fish in Alberta, especially our at-risk fish populations. And I think that's something that we need to start really prioritizing, so that we can actually understand where we should be focusing our efforts to help protect fish as we move forward from this parasite. And like I said, it's been almost 10 years. The whirling disease signs are still out there, but they're a little bit faded now. They used to be orange, and now they're more white. But this is the sign next to the Raven River that was one of the first places that we went and sampled for whirling disease way back. So we go there still to collect worms and catch parasites. But I think just an evidence that it's been a while. And with that, I'm going to stop, and I think I'll just maybe emphasize that the people up at the top here are all the folks that actually did the work that I presented to you. So they're the real drivers of the cool discoveries and all the innovation that we do. I'm the talking head that sits at the desk all day. And obviously this work wouldn't have been possible without support from Alberta Innovates, the Alberta government, NSERC, and Parks Canada. They've been really active and engaged participants in all the work that we've done. And some of the partnerships we've made while we've worked on these projects has been really great example of how academic, government, and industry partnerships can really advance our understanding of important issues like whirling disease. So with that, I'll stop. I think I'm nearly on time, which is rare for me. So I'm happy to take any questions. I'm going to stop sharing my screen. And
thanks.
There we go.
Thanks so much, Dr. Hanington. That was a nice, complete story, and it is good to know that as a 10-year anniversary is coming up, that we are much further along in what we know than we were 10 years ago. So kudos to yourself, your team, and your collaborators in advancing that knowledge. I want to thank everyone else for joining us today, and just remind you our next seminar is going to be on March 25th. It's Christy Morrissey from the University of Saskatchewan, and she's going to be presenting on advancing exposure assessment approaches to improve wildlife risk assessment, application to waterbird exposures in Alberta's oil sands. Again, just to remind folks, if you are not on the mailing list for these science seminars, please send an email to epa.ocs@gov.ab.ca to get added, just asking to be added to this list. And with that, one more thank you again, Dr. Hanington. Great presentation, great work. Look forward to seeing, well, I don't want to wait 10 years for the update next time, but we're learning lots. Thank you.